Hi group, and welcome to Unit 9. Today we're going to talk about the complement system. The complement system includes 18 plasma proteins that actually complement antibodies, and they aid in inflammation and lysing of cells. So complement is a group of 18 plasma proteins. These are free-floating proteins that are in the blood and made in the liver and sometimes in the macrophages that actually aid in lysing and inflammation. Okay, they actually complement antibodies, and primarily they are made in the liver, but like I said, sometimes complement is made in macrophages, and this is part of our natural immune system. There are four major roles of complement. Complement should promote an acute inflammation response. It should alter cell surface to encourage opsonization. And remember, opsonization is promoting the adherence of phagocytic cells, or when microbes are prepared for digestion and the bug is coated with molecules allowing them to bind with phagocytes. The third role of complement is that it modifies the cell surface, leading to cell lysis. And finally, the fourth major role of complement is to regulate humoral activity or humoral immunity. Let's go ahead and talk about the characteristics of complement. Okay, so they're plasma proteins, they're globulin proteins, and there is 18 globulin proteins that consist of the complement system. Some textbooks will tell you that there are 25 components of complement, but that includes the proteins or inactive precursor proteins. Complement is found in the serum of animals, and it is composed mainly of the globulin proteins, and these are mostly beta-1 and beta-2 globulins. It is a highly unstable compound, mostly found in the liver, especially components C3 and C9. And some complement components are formed in macrophages like C2, C4, and C8. And just so you know, complement is identified by the letter C, followed by a number which denotes its terminal sequence. Complement products resulting from a split in proteins during the activation process are followed by a lowercase letter, i.e., or for example, C3a, C3b, or C1q. These letters refer to the proteins of the alternate pathway. Now, sometimes complement has to be inactivated, especially when it comes to serological testing. For example, complement has to be inactivated in procedures like VDRL, which is the Venereal Disease Research Laboratory Test for syphilis. Okay? In serum, it is usually in the inactive form, but when IgM antibody responds, it gets activated. Activated complement can interfere with some serological tests and has to be inactivated. The primary way for inactivating complement is taking that complement and heating it for 30 minutes at 56 degrees. Okay? You can also inactivate complement by refrigerating it for 3 to 4 days, violently shaking the vial, anticoagulants, and certain compounds like acids, alkalis, ether, chloroform, bile salts, or soap. Those are all ways of inactivating complement, but the most common and best way to serologically inactivate complement is by heating it for 30 minutes at 56 degrees. Now, complement can be reactivated as well, and usually it can be reactivated by standing at 7 to 37 degrees Celsius for about 4 hours. Complement can also be preserved by freezing or lyophilization. In serological tests, we use complement to enhance some antigen-antibody reactions for visualization. Our usual choice of complement in the medical laboratory is guinea pig serum. Guinea pig serum is the usual source of complement in laboratory testing. It contains high amounts of complement and is more consistent than human serum. Complement is present in the circulation in the inactive form. It must be activated by antibodies or coming across a foreign substance. Okay? Complement activities are activated in 3 ways. Those are called the classic pathway, the alternative pathway, and the mannose binding lectin pathway. The principal complement components are C1 through C9. Complement is present in the circulation in the inactive form and must be activated when it comes across a foreign substance. It can be activated in 3 ways, such as recognition, when it senses its corresponding antibody, Amplification is the enzymatic process of activation. And then finally, the membrane attack unit, which leads to cellular destruction. The antigen and antibody reaction occurs in the recognition unit. Complement senses the Fc receptors on the antibody, and complement is activated, forming C1QRS. C1Q binds to an immunoglobulin molecule, IgM or 2 molecules of IgG, and then C1R and C1S activate the next step of the classical pathway. C1R and C1S don't bind to the immunoglobulin, but do activate the next step in the classic pathway. The amplification unit refers to the activation unit or the activation of complement factor C4. It is activated by C1QRS. C4 is activated by C1QRS, which is the most abundant factor in the complement system. C3, of course, is the most abundant. C4 is the second most abundant. C4 is activated and is broken down to C4A and C4B. C4B activates C2, C4B2A complex, which is also known as C3 convertase, promotes cleavage of C3 into C3A and C3B. C3 component, which is the most abundant component of the complement system, is broken down to C3A and C3B when activated. C3B then attaches to an RBC membrane, and the RBC is eventually lysed. Activated C3 leads to the development of the membrane attack unit, and the membrane attack unit includes complement components C5 through C9. Each component works to activate the next component in the system. It's actually amplified. C9 then attacks additional red cells and bacterial cell membranes by boring holes into it. The contents of the red cell or bacterial cell leak into the plasma, leading to intravascular hemolysis or bacterial cellular destruction. It is important to note that once complement is activated, each step that follows is more amplified. Now, the complement cascade order doesn't go in a order that makes any sense. It starts with complement component C1, and then it goes C4, C2, C3, C5, 6, 7, 8, and 9. The alternate pathway used to be known as the Propuredin Pathway because it was named after a protein once thought to be the first functional unit of the pathway. This is also known as factor P, the Propuredin Pathway. It is similar to the classic pathway, except there is no antigen or antibody reaction that is necessary to trigger this pathway. It's not initiated by immunoglobulins. It's always operating at a low level like a car engine idling. But the alternate pathway is activated by contact with a foreign substance like the coating of a microorganism. There are many components that are capable of activating the alternate pathway. Some components are capable of activating the alternate pathway like venom, inulin, which is a polysaccharide simple sugar produced by plants, and bacterial polysaccharides. The complement cascade can also be initiated by mannose binding lectin, which will bind to terminal mannose groups on a variety of bacteria. This is a cascade reaction similar to the classic complement pathway. C1 triggers activation of C4 and C2, but this pathway doesn't recognize bound IgG or IgM. It recognizes the mannose as a sugar on the surface of some organisms like Salmonella, Listeria, and Neisseria. Present in the largest quantities is C3, which is the major role player in the complement cascade. Both the classic and the alternative pathways cleave C3. This occurrence is the pivotal event in the final common pathway of both systems. The alternate pathway starts as C3. The alternate pathway bypasses C1, C4, and C2, so it does not require calcium, but it does require magnesium. Either way the complement system is activated, it'll activate the other system. Classic activates the alternate system and vice versa. Both activation methods result in cell destruction from C3 to C3b. You will want to know the physiologic effects and cellular consequences of complement. Okay? Complement components C1 through C9 will lyse red cells and lyse bacteria, while components C1, C2a, and C4b will neutralize viruses. C3a, C4a, and C5a play a role in inflammatory vascular response. They mediate hypersensitivity by generating anaphytoxins. C3a and C5a are potent chemotactic agents, while C3b and C4b play a role in opsonization. C1 through C9 will also affect immune complexes. So complement will alter the molecular weight, solubility, and composition of immune complexes. These are a cluster of large interlocking antigen and antibodies that are deposited in the tissues as granulomas. And then finally, C3a and C5a play a role in inflammation and the release of histamine. Complement is likely to be increased because of trauma or an acute illness such as myocardial infarction, which is a heart attack, or in certain inflammatory conditions. Serum complement levels are decreased in things like lupus, lupus-like syndromes, infections, immunodeficiencies, and genetic disorders. Specific complement deficiencies. Most of the time, people that have complement deficiencies, they either have Neisseria or lupus-like syndromes. That's something important to remember. But you can take a look at this list on your own. You can see that a C1R deficiency, your patient will have lupus, arthritis, or increased bacterial infections, and that goes all the way through C8. So feel free to look these over. You'll want to check this out. Now we want to regulate complement activity. This is called complement assessment. So complement can cause tissue damage if it's constantly being activated, and it can cause intravascular thrombosis. So we want to make sure that we evaluate complement to ensure that complement is not being used excessively or inappropriately by the body. Okay, so methods that we can use to evaluate complement would include complement lysing studies. Okay, will complement lyse cells like sheep cells? We can use ELISA to evaluate complement levels. We can use a liposome assay where we mix serum with liposomes, anti-DNP, and G6PD and NAD, and looking for enzymatic colorimetric reactions. And then finally, we can do radial immunodiffusion. Okay? Those are ways that we can assess complement. And complement is regulated by either inactivators or inhibitors. Complement is regulated by inactivators and inhibitors. Inactivators are usually enzymes that destroy the primary structure of complement components, while inhibitors combine with complement components in ways that prevent their further interaction with other intermediates of the cascade. They prevent interaction with other subcomponents. Complement is regulated by inactivators and inhibitors. Bad things start to happen to the body if complement is rarely activated or constantly being activated. A significant inhibitor is C1 esterase inhibitor. C1 esterase inhibitor, if you have a patient that is low on C1 esterase inhibitor, these patients have hereditary angioedema, which are fluid-filled swelling of the skin and itchy rash. Some complement inactivators would be C3b inactivator, C4 binding protein, C6 inactivator, membrane attack complex inactivator, factor I, and factor H. Okay, so a C3b inactivator is an activator that converts C3b and C3c and C3d and makes them inactivated. Okay, C4 binding protein inactivates C4b component. C6 inactivator inactivates C6. Membrane attack complex inactivator converts C5b to C5c and C5d. And then we have factor I, which the old name is C4b, C3b. Factor I is a little easier. And this factor regulates protein complements of the alternate pathway. And then we've got factor H, which is the H globulin, is the old name. It cleaves C4b and C3b proteins into inert fragments. Factor H enhances activities of factor I, and the alternate pathway directs complement to attach pathogens and not self-tissue. So some of our patients may have a deficiency of the complement system, and as you recall, deficiencies of the complement system can lead to all kinds of problems like lupus and Neisseria infections. Okay, so when complement levels are low, we have to consider asking ourselves, why are they low? Has complement been used excessively or activated excessively? Is complement currently being consumed, or is there a single component of complement that may be absent due to a genetic condition? So here's just kind of a way to outline a reaction involving complement. If you have an antigen, a specific antibody, and complement, it leads to lysing of that cell or lysing of that organism. But you have to have all three: antigen, antibody, and complement.